Good morning, everybody. Welcome to the second day of CCN. It's my pleasure to introduce the keynote for this morning. But before I do that, I just have a couple of logistics announcements. The first one is that there's a few limited tickets for the social available now at the front desk. So if you want, you can go to registration and get one of those while the supplies last. And then the second thing is that the food truck, please use it today because we have a minimum we have to meet, and we didn't meet it yesterday, so we're a little bit nervous now. Okay, so now I want to get to the exciting part. So today, we're very honored to welcome Dr. Nicole Rust to give a colloquium, a keynote. And it's very exciting to have Nicole back because she was one of the keynote speakers at the first CCN back in 2017. And today, Dr. Rust, Dr. Rust is a professor of psychology at University of Pennsylvania, and her research over the years has spanned from the basics of visual encoding to visual memory, and to higher level questions like how we should do experiments in neuroscience and how we should interpret the results of those experiments. And she's most recently written a book entitled Elusive Cures, Why Neuroscience Hasn't Solved Brain Disorders and How We Can Change That. In this keynote, she's going to focus on some of those issues, talking about what pieces might be missing to translate the progress we've had in computational and cognitive neuroscience into real cures for mental and neurological disorders, and how the field can bridge those gaps. What should the grand plan be for neuroscience research to achieve progress in these areas? So to help answer these deep questions, Dr. Rust wants to have a town hall discussion where everybody can contribute to talking about the challenges and how we think we can overcome them. So after her keynote, there will be a town hall, which will be led by Dr. Ivan Oransky. Dr. Oransky is editor-in-chief of The Transmitter and is a distinguished journalist in residence at New York University's Arthur L. Carter Journalism Institute, where he focuses on medical journalism. You also might know him as one of the co-founders of Retraction Watch, where he helps to flag papers that have been retracted to the rest of the field so that the field can progress more effectively. So he's someone who has this critical high-level perspective on achieving a practical understanding of complex systems to contribute to this discussion. So without further ado, let's dive in. Please join me in welcoming Dr. Nicole Rust.
So thank you so much, Andrew, for that wonderful introduction. Big thanks to the CCN organizers for the invitation to speak. And I'm grateful for all of you for showing up here at 8:30 in the morning. So today, what I'd like us to do is to zoom out. So way, way, way out, and really talk at this very high level about the big picture. And we'll be talking about brain and mind research at large, and just asking ourselves, how's it going? So in the first part of this event, I'm going to give a talk where I'll lay out my perspective and hopefully seed some thoughts. And in the second part of the event, we'll have a town hall that Ivan will lead, where we'll be really excited to hear your comments. You can ask questions, but it's not really about questions for me. You can catch me offline. I'll be here all week. This is really about comments and conversation. So what is this really all about? Well, Andrew's introduction actually was the big reveal of the title of my forthcoming book, and I heard some murmurs there. Yeah, it shocked me too. Insofar as those of you who know me know that I'm a foundational researcher, right? I study the nuts and bolts of how the healthy brain operates. And so some of you might be scratching your heads wondering, why did Nicole Rust write a book about medicine of all things? If you would have told me I was going to do that a few years ago, I would have been surprised to find out that that was true. So let me just begin by diving in and telling you why I was motivated to write this particular book. Like I said, before we begin, before I forget, do say that the issues we're about to talk about with regard to brain and mental disorders, they are complex and they can be a little bit sensitive. For example, there are questions related to whether some things we call disorders are actually forms of neurodiversity that society has wrongly become intolerant to. So cases of autism spectrum disorder, attention deficit hyperactivity disorder, and depression, those are all examples. When we use the terms disorder and dysfunction in this conversation, we're really using them with a very empathetic spirit. acknowledging that for a subset of individuals who have these conditions, they will hope for treatments. And we don't have treatments right now that are sufficient to help them. So it's with that empathetic spirit that we enter in this conversation. So getting back to the thread of why I wrote Elusive Cures. So yeah, I'm a foundational researcher. I study memory. I study questions like, when you have that percept that you've seen someone before, what's happening in your brain? And what were all the events leading up to that? So how was the memory stored in your brain in the first place? And the rationale behind foundational research of all types is that it's the first step. It's a real bottleneck in brain research to all sorts of benefits to society. So we want to understand brains to understand them, right? We're a curious species. We want to understand how we work. We also want to understand the brain for more tangible benefits, such as creating artificial intelligence, right, that mimics the intelligence of ourselves. And if we just take a step back and ask ourselves, how has this been going over the past decade? I hope that many of us will agree that the answer is just a sheer wow, right? New insights into how the healthy brain works, they're coming in like a fire hose. And in terms of the new AI we're building, like large language models, right, it's progressing much more quickly than I could have hoped or even dreamed. The issue is that if we think about another societal benefit, and that is understanding the brain to treat brain and mental illness, there are indications that that's not going as well, not only as some hoped, but even as some expected. And the purpose of the conversation today is for us all to just consider what might be going on there, and is there anything that we could do to help? To just give you a sense of some of the frustrations that are happening, let me read you this quote. It's the opening for a report that was published last year from the Wellcome Trust, which is a funding agency in the UK. Neuroscience has made impressive progress in advancing our understanding of the brain by leveraging cellular, animal, and computational models. Okay, that's us, right? However, while scientific advances have increased our understanding of anxiety, depression, and psychosis, little benefit in terms of modernizing treatment has yet accrued. This is a troubling disconnection. So let's just pause for a moment and just ask ourselves, is that really troubling? I mean, after all, the brain is held up as the most complex thing in the known universe. And anxiety, depression, and psychosis are held up as the most complex forms of its dysfunction. We've only really been at neuroscience as a serious effort for about 120 years. So we can ask ourselves, is it really that troubling that we haven't figured out everything yet? Maybe not. At the same time, if our response, not just to our funding agencies, but to society, is going to be, we haven't figured this out because it's complicated, perhaps we can unpack that a bit. Perhaps we can lay out what exactly is so complicated about it. And moreover, perhaps we can unpack a little bit our plans to tackle all of that complexity. So that's really what this conversation is about. It's about we, as cellular, animal, and computational researchers, laying out what are we most excited about in this era of brain research. I want to assure you that this isn't just about psychiatry, which we can all agree is really complicated. It's also true of neurology, too. Especially when you consider the neurodegenerative disorders, Alzheimer's, Parkinson's, Huntington's, multiple sclerosis, ALS. All of these are examples of disorders that we cannot stop the neurodegeneration. Consider for a moment Alzheimer's, where 30 years ago, researchers identified a particular genetic variant. If inherited by an individual, it leads to Alzheimer's later in life with near certainty. That's a gene called APP, and the protein is called amyloid. And in the individuals that are afflicted with Alzheimer's, that protein accumulates into clumps called amyloid plaques. So following that discovery, researchers in Alzheimer's very reasonably spent 30 years diving into the role of amyloid in Alzheimer's. And they even successfully created drugs that could clear amyloid plaques from the brain. It was heroic. Tragically, those drugs do not stop cognitive decline to the degree that we all really hoped that they would. And the big question is, what happened there? So we don't know the answer to that question. It's very nuanced and tricky. Carl Harrop, in his book, How Not to Study a Disease, The Story of Alzheimer's, lays out what he thinks might have happened there. And Carl in this book says something I find very poignant. It's whether we can admit mistakes, whether we can learn from our failures, and whether we can adjust our course and restart that determine whether or not we can be truly great scientists. So here, Carl. So the point of this conversation is really to take stock as a community and contemplate the course we're on, to ask ourselves, is it the best possible course in light of the end goal of treating brain and mental illness? I want to assure you that the goal of this conversation is not to eliminate foundational research, okay? It's not to turn us all into researchers who investigate disease. I started this project a foundational researcher. I'm coming out the other side a foundational researcher as well. It's really about conceptualizing what might be impactful in terms of that end goal, working backward, and asking ourselves, do we want to prioritize that? So I want to also assure you that it's not that nothing has been happening in the medical arm of brain research. It has. We have new treatments, some game-changing, to treat disorders like insomnia, migraines, postpartum depression, pain, and epilepsy, all built on the backbone of big breakthroughs in foundational brain research. The issue is that there are classes of disorders that we're having a hard time figuring out how to crack. They include the psychiatric conditions, depression, anxiety, and schizophrenia. They're all cases in which we do have some treatments, but the treatments don't work for everyone, and we don't understand what's happening in the brain. The neurodegenerative conditions, like I said, we can't stop the neurodegeneration for Alzheimer's, Parkinson's, or ALS. And the neurodevelopmental disorders, like forms of intellectual disability and autism spectrum disorder. We don't really know what causes them, and we don't really know what's happening in the brain. So the question is, how will we figure it out? So this all sets the stage for why it is that I wrote Elusive Cures. I've been in this field now, if you count my undergraduate research, for 30 years. And it really concerned me that I didn't have good responses to criticisms of my own field, like there are troubling disconnections. I didn't have a good concept of what all of us are doing and how everything fits together, and that worried me. I also couldn't find where this was written down anywhere. I assumed it was, and so I started looking for it, some white paper or some mission statement of a funding agency. And those do exist, but I couldn't find any that I was really optimistic about. One that I would read, and I'd say, yeah, that captures the genius of our era. That's why the brain's so complicated. That's what we're going to do to figure it out. We've got this, right? I didn't leave with that sense of optimism. And that left me a little bit pessimistic, because I felt like I couldn't really tap the genius of our era to guide my own foundational research. So this was very much a personal project for me, just trying to figure all this out. So when I set off to write this book, I set out with a question, which I did not know the answer to, and that is, what is the grand plan for brain and mind research? And by grand plan, I refer to that very affectionately. It's what I see as a description in broad strokes of how we, as a community of brain and mind researchers of all types, plan to get from where we are now to societal benefits of all types, which include but are not limited to better therapies for brain and mental dysfunction. And as I see it, the grand plan is not as abstract as figure out how the healthy brain works, figure out what causes its dysfunction, develop treatments. That's a sketch, that's not a plan. What's our plan of our generation to actually do those things? I also want to emphasize that I think the grand plan is much bigger than any one of us. It is the genius of our community, of which I am but one member, and you are all but one members too. When I approached this project, I did not approach it as, I'm going to be the genius that's going to come in and figure all this out. It wasn't like that at all. It was much more about channeling the ethos of our era, learning about what all you are working on and how it all fits together, right? And what's going to come out of it. That's what it was about. And so that's the big question today. What is the collective genius of our era? What is our grand plan? So that's a really hard question to answer. It took me a really long time to answer it. I wrote this book, I think, like 3 times or something, right? So I would write it, and I would say, nope, that's not it. And I would write it again, nope, that's not it. And it took me a long time to converge on something that I felt truly optimistic about. So I think that just to set the stage, it helps to rewind a bit to the 1990s, where the grand plan was much more obvious, and then just ask ourselves what went wrong there, and what are we going to do about it? So in the 1990s, researchers in the brain research community really rallied behind 2 technologies, genetic sequencing and non-invasive measures of human brain activity, fMRI and PET scans. And the general idea was that if we just sequence genes and image brains, insight into how the brain works and what causes its dysfunction would naturally follow. So just to give you some context for that era, it was the era of the Human Genome Project, which was launched in 1990. The goal was to sequence the composite genome of one individual. And it cost, I think, around $3 billion and took 13 years. When researchers went into the Genome Project, they knew that C. elegans, the little roundworm that many of our colleagues study, with its 302 neurons, had 20,000 genes. And one question of great interest of that era was, how many genes do humans have? With all of our complexities and our 86 billion neurons. So a fun betting pool was started called the Gene Sweep to collect guesses. The mean guess was 62,000. The maximum guess was 200,000. The lowest guess of all of the researchers who participated was 26,000. The result of the Human Genome Project revealed that we have only 25,000 genes. C. elegans, with its 302 neurons, has 20,000 genes. We, with our 86 billion neurons and all of our biological complexities, have 5,000 more. It blew everybody away. Now, since then, we've revisited that number, knowing that it's no longer one gene goes to one protein, but one gene leads to many proteins due to modifications at the transcriptional and translational stage. But of this era, that was the answer. And so naturally, researchers wondered, maybe our complexity is massively reducible to simple things like genes, right? So that's the ethos of this era. And you can still see vestiges of that, including in the undergraduate textbook I teach from, which figure 292.2 lays out a plan for how we're going to get from genetic variation to new treatments for psychiatric conditions like schizophrenia. It begins by finding a gene whose variation is tied to the disorder. You then mimic that mutation in a mouse, use that mouse to study it, you create a drug, put it through clinical trials, and voila, we have a new treatment for schizophrenia. It's called molecular medicine. And there was tremendous optimism about molecular medicine not so long ago. Today, we regard this approach, at least stated in this simple way, as a bit naive, because now we know that psychiatric conditions like schizophrenia and others are not tied to variation in individual genes in all but very rare circumstances. They are tied weakly to variation in hundreds of genes, and there are strong environmental effects. So this plan, as stated in this simple way, is just not going to work. So another thing we were really excited about in this era was non-invasive imaging of the human brain. And it's obvious, right? If you could just look in there under the hood, you could figure out what's happening in the brain when an individual undergoes a psychotic episode or when an individual falls into a bout of depression. This enthusiasm was channeled by one of the pioneers of that era, Nancy Andreasen, in her book, The Broken Brain, published in 1984. As they improve and become more accurate, these imaging techniques and other laboratory tests for mental illness will become part of standard medical practice during the coming years, thereby improving the precision of diagnosis and assisting in the search for causes. It's 40 years later, and that still hasn't happened. It's still the case that many brain and mental disorders that we were hoping would be diagnosable with a genetic test or a brain scan still cannot. That's not to say we haven't learned a lot from genes and imaging brains, right? We have. And it's not to say that these technologies will not be part of the solution. Of course they will. But I'm guessing that most of us will agree that we're no longer really optimistic that if we just sequence genes and image brains, everything's just going to work itself out, right? So the question is, well, if that's not our grand plan, if our grand plan has changed, what does it change to, right? What is the ethos of this era? One way to approach that is to get excited about all the new technologies we have that they didn't have in the 90s, right? We have optogenetics, we can manipulate brain activity with unprecedented spatial and temporal resolution. We have CRISPR to edit genomes. We have all the omics. We have genomics, transcriptomics, proteomics, connectomics. We can record from neurons at large scale, or up to 1 million simultaneously recorded neurons in a mouse. We have machine learning to help us sift through all this data. We have large language models that are giving us insights into what intelligence is and is not. I think it's really easy to get caught up in the enthusiasm that these big discoveries are imminent. But we might also take a step back and say, well, what is the plan to do? What are we going to do with all this data, right? What are we thinking about? One might even suspect that what we have going on is a little bit of chaos in the brickyard of brain research. So that phrase comes from this lovely parable published in 1963 as a letter in Science. And the author likens the scientific enterprise to building large, imposing edifices, and the data we acquire in our experiments as bricks. The parable begins that in the beginning, scientists would construct their edifices by commissioning specific bespoke bricks to minimize waste. We then transitioned into an era where we just collected bricks into big piles with great pride. And sometimes we couldn't even build edifices anymore because we couldn't find the right bricks in the big mess. And the parable ends, saddest of all, sometimes no effort was made even to maintain the distinction between a pile of bricks and a true edifice. The point of chaos in the brickyard is that there are two things that need to happen for us to achieve our end goals. We need not just the data, we also need blueprints, right? So in the 1990s, our bricks were genetic sequencing and fMRI. We now have a whole bunch of other bricks, but what are our blueprints, right? So let's talk about what the blueprints were in the 1990s. Those were laid out in this brilliant paper written by Eric Kandel, the Nobel laureate, in a paper called A New Intellectual Framework. In that paper, he lays out 5 principles to support the framework. Kandel is channeling the ethos of his era when he wrote this paper. And when I unpack it from you, I think it's going to sound very familiar. He wrote the paper for psychiatry, but it also applies to neurology as well. So it all begins with genes. Genes are translated into proteins. Proteins combine into neurons. Neurons are configured into neural circuits. And it's the activity of those neural circuits that gives rise to all of the brain's functions and its dysfunctions. In addition, our brains are shaped by our experiences, where behavior and social factors feed back, in Kandel's framework, to modulate the genes that are expressed, which in turn determines how all the neurons are connected together. That happens via learning. Kandel emphasized this arrow here, how the brain gives rise to the mind, is the great challenge for biology and psychology to delineate this relationship. And many of us in this room have been working really hard in the intervening period to try to figure out what's going on there. Many of us, in the framework of thinking about the brain as an information processing machine that takes information in through its senses and transform that information as it propagates through the brain, through a series of modules like the visual system, the auditory system, attention, memory, decision-making. And because there's been so much progress in working out what's going on in that era, well, let me just first give you an example. One example of this arrow, which we often call this level algorithm, is a deep artificial neural network model of the visual system. So because we've been working so hard, I think, on this arrow, and I want to include it too, I'm going to call this the molecular neuroscience framework extended to incorporate the algorithms that Kandel didn't really lay out. So in the process of writing the book, I sifted through an enormous literature well outside my wheelhouse, right? So not just the types of things I work on, vision and memory, but functions and dysfunctions of all types. So depression and mood, also motor function and paralysis, disorders of consciousness, coma. And one thing that really struck me is how the degree to which over the past 30 years, we have really been focusing on how the brain works by describing how causes lead to effects. So how the expression of proteins affects a neuron's function, or how brain activity leads to behavior, or in a deep convolutional neural network, how what's happening in V1 triggers what happens in V2. And so this makes sense, right? So if you task brain researchers, foundational brain researchers, describe how the system works, descriptions of how causes lead to effects, they're very satisfying. We've all done it, right? You set up the dominoes in a row, you tap the first one, they all fall. Naturally, if you think about the brain this way, if you're thinking about the causes of its dysfunction, it leads you on the path of looking for broken dominoes. So it's what I call find the broken domino and fix it. I've already shown you an example of this. Find a gene, make a mouse, create a drug. That approach actually it's celebrated with its own name. It's called rational drug design. One gene, one drug, one disease. But it's not just happening at the genetic level. We're also doing this at the level of brain activity, where we're looking for brain areas that have aberrant activity so we can go in and stimulate them to recover their function. And in some cases, this approach has been quite productive. For example, one treatment for depression involves stimulating prefrontal cortex based on the observation that in the prefrontal cortex of individuals with depressive symptoms, activity is a little bit lower. Okay. So I'm not arguing that this is everything that's been happening in brain research over the past 30 years, but my read on the literature is that this has been a very dominant, and I would even argue the predominant approach. These are the blueprints of the 1990s, and they've carried over for some decades. And so insofar as we want to question, like, are these the right blueprints, we might want to ask ourselves, is there anything missing from this slide or anything wrong with it? One thing that I find incredibly notable is the increased emphasis in recent years on these feedback loops. We're seeing it at all levels, right? We're seeing it at the algorithmic level, where our feed-forward neural networks are becoming recurrent, so CNNs are becoming RNNs. We're seeing it at the level of genetic regulatory networks, where there's increased emphasis on how complex feedback loops lead to the regulation of gene expression. We're seeing increased sophistication on questions about how the environment feeds back to shape how our brains are wired up. And that you can see, for example, in the newest generation of models of reinforcement learning. And so why I find this so exciting is because I think it's absolutely game-changing. This isn't just another technology that adds another brick. It's not just another domino in the chain. When you have systems where causes lead to effects, that feedback is causes, those systems behave in different ways. And their dysfunction also might be different. These are what we call complex dynamical systems, and their behavior often has surprising properties. We often call that emergent. So the classic 101 example of an emergent property is the interaction, the oscillation that emerges in a predator-prey ecosystem. So lynx and rabbits. Lynx eat rabbits, and so the number of lynx around is going to depend on the number of rabbits around to be eaten. But as lynx deplete their food source, their number of lynx begins to fall. And then the rabbits recover, and then they rise again. And so what you get is this periodic oscillation of these two populations. The important insight here is a very simple example of how the emergent property, the oscillation, it emerges from the interaction between the two nodes. It's not what's happening in each one of the dominoes individually. We have every reason to think that in biology, we're going to see lots of examples like this. One example is blood pressure, which we don't think of as a broken domino, but we think about a system set to the wrong point. So the way blood pressure works is there are pressure sensors in your vessels that send information to your brain. Your brain compares the pressure of where it is with what it should be. And when it's off, it sends feedback signals back to your body to adjust. So increase your stroke volume of your heart or increase the diameter of your blood vessels. When an individual has chronically high blood pressure, we don't look to see where the broken domino is. What we do is we think this is a system that's miscalibrated, and we need to recalibrate it. And so we'll give them drugs to do something like increase the diameter of their vessels, even if we haven't determined that that's the problem in the first place. So my punchline here is that I think one of the big bottlenecks that's been happening is we've been focused so much on the brain, treating it like a domino chain, and that's not the type of thing that it is. My prediction, thus, is that as we shift to thinking about the brain as a complex dynamical system, as so many of you already are, the shift is going to be really impactful. So let me just break down a little bit why I think that, right? It's easy to say that, but why would you think that? For me, thinking about the brain as a complex dynamical system really illuminates the formidable problem that we're up against with the end goal of creating treatments, right? So instead of trying to figure out how the brain works and treating it as broken dominoes, we're going to kind of fast forward to the end and then try to work backward. And just to illustrate this, I want to spend a few minutes with this friendly equation. It's called a logistic map. Some of you may have seen it before. It's the simplest possible thing, right? It's just one thing. It's x, and it's a little recursive equation. So the value of x at the next time point, x sub n sub plus 1, is computed as the value of x at this time point, x sub n, times 1 minus that, multiplied by a scalar. This little simple equation has some of the most amazing and complicated behaviors. So let me just begin by showing you what happens in a reasonable regime, right? So if we start x sub n with an initial condition of 0.2 and the scalar at 1.5, it starts at 0.2, it reaches some set point, and it just stays there. Great. But now, let me show you what happens if we increase r to 3.2. My guess would have been that it would just increase to a higher set point. No, it doesn't do that. It begins to oscillate. Okay, so this is an emergent property. Now let's increase it a little bit more to 4. Now you see that the behavior of this simple little system becomes aperiodic. It actually becomes chaotic, meaning that if we now change our initial condition from 0.2 to adding a little tiny intervention, 0.00001, you can see it does the same thing for a while, and then its behavior diverges. So this little tiny perturbation had a big unpredictable effect. This is what Edward Lorenz coined as the butterfly effect, right? It's the notion that a butterfly flapping its wings over Brazil can cause a hurricane in Texas. And what it suggests, right, is that in these complex dynamical systems, right, little perturbations have big effects. So what does this have to do with the brain? Well, if we just take a step back and again, fast forward to the end, it's important, I think, to conceptualize what we're trying to do. So we're working with a system, whatever system it is, the mood system, you know, the balance system that leads to an epileptic seizure. And what we're trying to do is shift it from an unhealthy to a more healthy state. That's what a treatment is about, right? And that really amounts to a form of control over a system. And I don't mean control in the creepy sense. I mean control in the sense of shifting a system from one state to another. You might want to call it influence. Insofar as what we're dealing with here are complex dynamical systems, we know they can be exceedingly difficult to control. They don't have to be chaotic for this to be true. When you have a system where causes lead to effects that feed back as causes, perturbations to the system reverberate. Now, our community already knows this. We've seen this in the era of optogenetics, which we were so excited about out of the gate, right? We can finally causally perturb systems. And then we started doing those experiments, and they made no sense. And the reason they made no sense is because our models were basically domino chains. They were oversimplified. And so these perturbations just had these big, unpredictable effects that nobody knew how to interpret. We also see this in cancer therapy and other types of therapy, where a cancer could be initially sensitive to a drug, and then it gains resistance to it. And it does so because there are all these feedback, complicated regulatory loops. And so when we block one path, the cancer just finds another. A non-biological system is, of course, the weather. As we all know, we cannot predict the weather very far out. This is because we can't measure the initial conditions, the conditions right now, with enough precision to accurately predict what will happen far into the future. One of the things that's been a bit lost in the history of weather research is that our initial goal wasn't just to predict the weather, we actually wanted to control it. And that makes a lot of sense, right? A hurricane's coming, it's going to cause lots of death and destruction. We want to shift it or maybe dissipate it. We tried hard. In fact, there were a few disasters. And it turned out we realized, you know, the weather really can't be controlled. At least we never figured out how to do it. And so I think one big question we might want to ask ourselves is what makes us think the brain will be more controllable than the weather is? I think there are good answers to that, and we're going to unpack them in just a moment. But I think that instead of assuming that the system is simple and that its dysfunction can be attributed to broken dominoes, this might be a much more productive approach. Assume that it's near impossible, and then face that complexity head on, right? And that's the way we're going to tackle this. So before I unpack how I think we're going to go about this, I just want to take a moment and reflect on this kind of shift that I see happening in terms of the grand plan. The 1990s, the bricks were fMRI and genes, and the blueprints were domino chains. And the grand plan might be summarized in terms of something like find the broken domino and fix it. I think we can all agree that in the 2020s, we have shifted our bricks. We now have lots of different things. We have CRISPR, we have omics, we have all large-scale brain activity. I see the blueprint shifting too, and I find that really exciting. And insofar as we're all on board with the idea that the brain is a complex dynamical system, those are the blueprints, the question is, well, how would we summarize the grand plan? My sense is that it's something like we want to acknowledge that that's what the brain is, and we want to model it in that way in order to infer its fragility, so figure out where its dysfunction is happening, and determine how to control it. Not in the creepy sense, again, we're not trying to mind control or anything like that. Again, just to shift it from those unhealthy to those more healthy states. That's how I would describe our era's grand plan. And just to reflect on the shift, I just want to bring up this famous quote by George Box that so much of us love. All models are wrong, but some are useful. So when Box uttered this phrase, wrong in the sentence refers to the fact that all models by design are meant to be simplifications, right? We're not trying to recapitulate all of reality. We're trying to recapitulate some fraction of reality that we think matters. We might think of what we were doing in the 1990s and this emphasis on domino chains. There was nothing wrong with it. There was nothing inaccurate about it. We were mapping out the domino chain backbones in these systems. However, that simplification that ignored the feedback loops, ignored the fact that the brain is a complex dynamical system, it made those models not just wrong, but also not useful for illuminating some types of functions and their corresponding dysfunctions. The hope is, as we shift to thinking about the brain as a complex dynamical system, incorporating those feedback loops in again, we're going to make models that are still wrong, because all models are wrong, but at least the models will become more useful. So I just want to end this section by focusing on, insofar as we're on board with the idea that our goal, our grand plan, is to infer fragility and determine control to shift the brain from unhealthy to more healthy states. And we agree that that's a really hard problem to solve. How are we going to solve it? Like, what kind of conceptual approaches? Everything I've said so far is very abstract. So let's try to make it a little bit more concrete. And so the first way, I can think of two ways, right? The first way really focuses on trying to figure out the system's control modes, whatever system you're working in, mood or brain balance, whatever it is, motor function. It's a very engineering way to think about how to solve the problem. It's how engineers think about controlling the International Space Station. So the ISS orbits around the Earth, and it is comprised of a number of modules that are held together. And it is a complex dynamical system whose parts, the modules, are affected, they interact via physical forces. And the way that the system is controlled is via thrusters that are applied to each module that need to be coordinated so the ISS does not break apart, okay? And that's controlled by a model of the ISS. And so when engineers are trying to figure out how to intervene or control the ISS, they have to traverse an axis between models that are too simple, and so the interventions that they determine will be ineffective, or they could even be harmful. They could rip the ISS apart. And models that are too complicated, so interventions cannot be computed or computed quickly enough to, respond to an event. And so there are going to be some facts about the ISS that don't matter. Who cares what color it is? That doesn't matter if you're trying to figure out how much to thrust a thruster. But there'll be other things that do matter a lot, and then there'll be some that matter just a little bit. And that's the curse of complexity, like figuring out where to cut that off. We might get some insight into how to think about this in biology from cancer researchers who think about leukemia. As like other cancers, it's a problem in which cells have they've shut off their normal cell death and they're over-proliferating. So the ambitious therapeutic goal is to turn normal cell death back on again. And so they look to these very complicated genetic regulatory networks that are involved in the cell proliferation and cell death. Not knowing which ones matter and which ones don't, they just model them all, okay? So this is a model for leukemia. It has 62 different genes that interact via 140 different interactions. But once they have this very complicated model, they then go through a process of model reduction to reduce it, eliminating all the things that don't matter for controlling normal cell death. That leaves them with just 5 genes, a domino chain backbone and to regulatory controller proteins, and now they have these obvious therapeutic targets. This type of approach is also being applied. It's been used to create a new drug for Parkinson's, which is now undergoing clinical trials, and it's also being used to approach new ways to think about Alzheimer's disease, not just at the genetic regulatory level, but thinking about how genes interact with other components of cells and even organ systems. So that's one way we might think about what we're doing with all this data, right? We don't know what we're going to need to know for the end goal of control. So why don't we just collect a whole bunch of data, create really complicated models, and then we're going to reduce those models to figure out what it is that we do need, right? I think it's a rational approach. Another way to approach this problem is to say, well, that's going to take a lot of work, and in some cases, we can probably make a good guess about what the brain's control systems are. One example is the arousal system, right? This arousal system exists to kind of wake the brain up when something really important happens. It involves the neurotransmitter norepinephrine. It's an obvious control system in the brain, and one can imagine that we could just target that directly for certain types of dysfunction, like ADHD. So in both of these cases, the emphasis is on finding the brain's control nodes, right? And the assumption is that the brain is different than the weather because it has evolved these natural control nodes, and it's those control nodes that we're after. I also want to highlight that this approach might not always work, right? In some cases, identifying the control nodes might not work, not because the brain is uncontrollable, but because it would require adjusting the expression of hundreds of genes or the activity of millions of neurons. And the question is, we can't reduce it to a control node. So the question is, well, what are we going to do then? I think there's a good answer for that, and it's one I'm imagining might resonate with the CCN community a lot, and that is, these are cases where we might think about enhancing the innate plasticity of the brain to reshape neural circuits. So the idea here is that, there's a natural healing process that our brains always tap into, following a stroke or following when we experience a traumatic event. But in some cases, we don't achieve complete healing, right? And so the idea behind this approach is that we want to enhance that natural healing process, rely on the brain's natural process for rewiring itself. One example are rehab type approaches, where this is a non-invasive brain-computer interface that reads signals out of the brain and then teaches someone who's had a stroke how to tap into those signals to recover motor function. While speculative, there are also ideas that most of our brain-based interventions for depression, this is actually how they work. TMS, vagus nerve stimulation, exploratory therapies for DBS, and even antidepressants. They are thought, and there's some evidence that they work by enhancing plasticity. So the brain has become entrenched in a depressive state, and there's a plasticity that's stimulated, which the brain then can kind of wiggle itself out of it. And of course, we have behavioral and talk therapy, which is always, trying to tap into those natural learning mechanisms to reshape the brain. And a lot of exploratory therapies right now are trying to figure out how to enhance that process with drugs. This is kind of the speculative idea behind psychedelics. The thing I find fascinating about this approach is it just acknowledges the brain is really, really complicated. So complicated, we are probably not going to have any type of technology to deliver the intervention we need with the precision we need anytime soon. And so we don't want to think about doing that directly. That's not our therapeutic target. Our therapeutic target is to work with the brain's innate plasticity. So I just want to circle back and end on a kind of a personal note. The reason I wrote Elusive Cures. So like I said, I'm a brain researcher. I've been doing this for about 30 years. And I very much think that progress in brain research, it's about all the discoveries that happen down here, right? It's about every poster at this conference and all the hard work that went into all of that. However, when we do those projects, we have to decide what is it that we're going to work on, and we have to decide how are we going to approach it. And this is where I think we can benefit from thinking up here a little bit before we dive in. For me, even though I've been in this field for 30 years, I really didn't have a good sense in how everything fit together. And so I'm really grateful that now I do. I entered this project a little bit pessimistic, to be honest, and I'm not anymore. I'm really optimistic about what's going to happen in the near future in our field. I can answer the criticisms like troubling disconnections. I can tell you what's so complicated about the brain. I can tell you what we plan to do about it, and we don't know until we try. But I can also tell you that I'm really optimistic about what's going to happen. I couldn't find any good responses written down anywhere, so I wrote them down for anybody who wants to dive in a little bit more. And I'll just end on this personal note that for me, thinking up here, it's completely transformed everything I'm going to do down here, right? Like I just have a different concept of how the brain works. I have a concept of what matters about it. It's really had a big impact on me. So in the last few minutes, I just want to make now this transition to the town hall, where I'm really excited to hear your thoughts. And I don't necessarily expect or even want everyone to agree with my take. Like this is what I think, right? But what do you think, right? It's a diversity of perspectives. I want to highlight that there is a lot that needs to happen. Like I've just focused on one slice because I feel like it's the slice that nobody is kind of laying out very clearly. And it took me a long time to kind of piece that bit together. Again, the ethos of our era. So just to lay out some of the conceptual space as we transition to the town hall, I find it, any one of these problems could be its own book. But I find it helpful to conceptualize them by putting these things in different boxes. I've talked about this box over here, which I think can be traced back to the foundational challenges. And I would conceptualize it as saying, what we discover about the brain depends on what we think about it, the brain and mind, and what we're looking for, right? So if we look for domino chains, we'll find them. If we look for dynamical systems, we'll find them. That's certainly not the only thing that needs to happen. Another big one is precision medicine. So let me just quickly unpack this one, because if you asked a lot of researchers what's the big bottleneck, they would point to this one. So precision medicine is the issue that it comes from cancer and other disorders like cystic fibrosis. It's the discovery that the same symptoms can have a multitude of underlying causes. Like many different genetic variations, mutations can lead to the same symptoms. And the issue here is that it creates a chicken and egg problem, right? So how do you figure out a cause if you can't even group together people with the same cause? There are entire programs, for example, at the National Institute of Mental Health, called the RDoC program. They're really focused on how do you tackle this problem. And one suggestion is that what you do is you group all together all of those individuals with similar symptoms, and you measure a bunch of stuff about them, their genetic variation, their brain activity, their behavior, their life experiences, and then you use that to somehow cluster them into different clusters with shared causes. Each of those causes you can treat, okay? So this is a real problem. It's a problem of individual differences. And what I see as our role as foundational researchers is to help contribute to those measures. You can't just say measure brain activity, right? We're trying to figure out if you're talking about something like mood and depression, what is it that you want to measure about the brain? So that's precision medicine. There are also a whole bunch of challenges related to benefits to society. And I really don't want this town hall to be all about medicine. I think any benefit to society is worth talking about. What are our challenges and how are we going to overcome them? In the case of medicine, everything in this box really has to deal a lot with clinical trials, which are expensive, and it's rare that a drug makes it through, and so on. And in the final box, I will put systemic problems that are issues across all arms of research, and these are also big problems that we need to solve. They include things like failures of our findings to replicate and data falsification. Ivan has been so great about helping contribute to solutions for some of those issues. Other things like incentive structures, right? We have to publish sometimes quantity over quality, scarce and fluctuating resources, and that sort of thing. Okay. So those are the things I'm eager to talk about. And anything that comes to mind. I guess I should also say there's this last one, AI. And I think if you ask people, what are you excited about? They say AI. And then, so the question I would ask is, we're all excited about AI, but what do we expect AI to do for us exactly, right? It's another tool. So what do we want there? The final note as we transition to the town hall, we have asked that the video not just be pointed to us, but the video be pointed to you as well, the questioners. So we're going to have this microphone that you'll come up to. And so we were asked to just announce that if you come up, you will be videotaped. And if you're in the line of sight of the video camera, which is over there, if you're behind the speaker, your likeness might appear on video as well. So with that, I'm going to just leave this slide up, kind of the conceptual space, and I'm going to hand things over to Ivan so he can lead us through the town hall. Thank you very much. Test? Yeah.
Yep. Great. Well, it's really wonderful to be here. It was wonderful to hear that. Nicole and I have talked about this a bit, and so I had, there were some things that weren't surprising, but actually still both surprising themselves and then sort of my reactions to them also. And so really what we want to do in the next not quite 5 minutes is have a conversation. This is a town hall. You're going to have lots of them in the next few months for other reasons, but I hope that this town hall is going to be, focused on science, on progress in some way, maybe not necessarily on progress, but on a plan for progress, and also just a frank exchange of ideas and all sorts of, again, conversation. And I want to start because I want to give everyone time to think through how they might want to contribute to this conversation, to this town hall. I'm going to start with a couple of reflections of my own based on my own experience and all of that, and hearing what Nicole had to say. I took lots of notes, even though I had discussed this with Nicole earlier. And so again, there's a microphone right there, and we really are looking for this to be a conversation. Nicole and I could have I'm sure we could talk for 45 hours, let alone 45 minutes. It's not really the point. So the less we're talking, the more successful, I think we would agree, this is. So please do, again, they're right there. Please step forward, and you'll even have a minute or two while I gather some thoughts myself to share. And there were a couple of high-level themes that I wanted to reflect on and sort of respond to a little bit as we're gathering. And one is, again, based really on my own experience. And so none of you would have any reason to know this, and it's not exactly sort of anything I do anymore, but I actually trained in psychiatry. So I went to medical school and did at least part of my psychiatry residency. And in listening and in thinking about this and talking to Nicole, one of the things that I was reminded of is that, and this was in the mid-1990s. I mean, I know I don't look old enough to have graduated medical school then, but that's when it was. And one of the things that I was reminded of was, you all are probably familiar with, or at least have heard of the DSM, right? The sort of quote-unquote Bible of psychiatry. We could have days of conversation about what's wrong with that model, but it has criteria, it has diagnostic criteria, that's what it does. And one of them in the 90s was for psychotic disorders, was that if a particular patient you were seeing responded to antipsychotics, that was a criterion for having a psychotic disorder. I want you to think about that for a second, right? Some of you may be familiar with something called the Texas sharpshooter fallacy. Does that ring a bell for anyone? A sort of statistical metaphor or concept, with all due respect to people from Texas. It's not about you. I mean, it probably partly is. So this is, you have, and it used to be sort of the kings would shoot arrows at things, and then they're, or in the case of Texas, probably people are shooting other things, and they wouldn't have a target when they set up, but they would run over to the, to the wall or the side of the barn or whatever it was that people were shooting and draw a circle around it and say, oh, look at that. You've just hit a bullseye. So this notion that we must understand the way the brain, the mind, whether it's circuits or a particular neurotransmitter or anything like that, we must understand how it works because we've created this drug that we actually don't understand how it works. And then we've decided that if people respond to that, they must have this disorder that we were trying to treat in the first place. It's pretty interesting, right? Also logically problematic. So maybe I'm delighted that there are already several people standing here, so I'm going to shut up, and we would love to hear from all of you. So please just introduce yourself, whatever you want to say about yourself, and then we'd love to hear from you.
Hi, my name is Daniel. I am somebody who is on the autism spectrum and has ADHD, and in my experience, in reference to the feedback loop, so the interventions that have worked the best for me haven't been medical related at all. They have been completely external. And so one of the biggest, areas of concern for me as somebody who has read the DSM and multiple editions behind to figure out what the heck the history of my own disorders is how even with the feedback loop as you defined it, I think the biggest part of the feedback loop, especially for the brain, is purely external. It's whatever gets input. Just as an example, growing up, I was a very sedentary kid with a lot of respiratory issues, and I was obese, and instead of being assigned an exercise routine, I was assigned Prozac. The exercise routine as an adult has helped significantly more, and so I guess I was really curious about what the direction of medicine, especially neuroscience and psychiatry, has to say about more, environmental medicine, prescribing people routines and habits and environments instead of medications.
Yeah, thank you so much for that comment. I think there's a lot of conversation right now about should we target the brain, the mind, the environment, all three? And it seems like a holistic approach isn't the obvious one, right? And I was hoping to make that clear that I'm a proponent of that on that last slide, right? By the last type of therapy I talked about was talk therapy, right? Behavioral therapy. I think it's clearly going to be part of the solution, and it makes sense why that's part of the solution, right? And there are going to be nuances between disorders, right? I mean, behavioral therapy probably isn't going to help us a lot from stop the neurodegeneration associated with Alzheimer's, right? So different disorders are going to be different. But thank you so much for sharing that experience. That's really helpful and important.
Thank you. And I just want to, again, just to pepper and maybe have people think about this a little bit. Talk therapy, of course, is something else I was trained in, but the environment, right? And so how do we think about that when we think about feedback loops, right? It's one thing to say, well, you do this and you perturb this, circuit or it oscillates, but then you walk out and I don't know, it was raining a bit when I got here and I don't know what it's doing now, but tomorrow it won't be. I'm subject to noise depending on where I am. I'm subject to all sorts of pressure, stress. How are we accounting for that, right? So, please.
Hi, I'm Jill Crease. I'm a postdoc at Stanford University. And one thing that I am concerned about is, I think, like a systemic problem that the big journals like Nature and Science often publish new fundamental research, but like disease research at a more functional cognitive level is often not seen as something that is worth publishing in these big journals, which again drives researchers to that are ambitious to move away from disease research.
Is the system set up in a way that allows for this kind of exploration? Is it set up for a grand plan, or does it have a grand plan of its own that we're all just sort of hanging out in? Yeah, please.
Okay, thank you for the infectious optimism. in the end. So I want to, one discussion, one thing that kind of keeps me up at night is this, the idea of precision medicine and whether we can make progress there. And, you know, obviously we have made immense progress at mapping individual brains. We can take brain scans, we get a good understanding. And I think if we learn one thing is that there's tremendous inter-subject variability in how things are laid out. And that's even in the healthy population, well-functioning population. So it's like trees in a forest, right? I mean, every tree has a trunk and some leaves, but right now, the key thing is that we are mapping somehow how exactly the branches and the leaves on each tree are arranged. And the question is this really, so how do we distinguish between biological and functional variability that matters for function and that doesn't matter for function? And that's especially a problem, I think, for AI research, where we take these big databases and pipe it into some machine learning algorithm and hope that we're going to get something out in the end. Like, is there, do we actually stand a chance right now with the data that we have, given that we don't really understand how that variability is arranged and organized? So that's kind of, I just wanted to raise that, get your thoughts on that.
It's such a crucial point. And I'm just curious, as a show of hands, how many of you in this room, something about your work really captures individual variability and tries to kind of explain it? Is there, yeah, okay. So I'm glad to see so many hands go up, right? We're working on it. But yeah, do we have the right data? And what kind of data do we need? Let's dream big and try to think about what would it look like? If we had all the data, what would we do with it, right? Yeah. I don't have answers, but I think these are great questions.
Yes, so my name is Nadia. I'm from Brown University, a postdoc. I'm wondering, we have been talking about feedback loops and the RDoc initiative, and we mentioned that we want to help, bring the puzzle together. But I'm also wondering whether it's our responsibility to point out certain flaws in that approach or certain possibilities to make it better. I'm thinking about symptoms and how they are measured and how they are often multidimensional. And it's very important for modelers who are maybe not in a day-to-day interaction with patients to understand what are the implicit assumptions or information that a clinician picks on that isn't reflected in these clinical questionnaires. And I think a lot of information gets lost there, and that sometimes makes me a little bit pessimistic of how good can my model be if the input is only that precise. I might not need to change my model, I might just need to have more precise inputs into the model. And with respect to that feedback loop, I'm wondering, we have been talking about using treatment response to categorize disorders, but what about using treatment responses to actually look into individual differences, that those who are responding similarly to a medication are grouped together? And I would see that as a feedback loop to our RDoc initiative, but I have to say that one of the challenges I face with that approach is that it's not fundable, if you want to go in the other direction.
So much to there it goes. I think the thing I would highlight is, as we shift from thinking about the brain as domino chains to thinking about it as a complex dynamical system, we actually do need to rethink every step of the scientific process, right? Which starts with measurement. So we have to describe what it is we're trying to explain. That's what you're talking about. And it does have a big impact on how we go about measuring things. For example, when the parts are interacting, you have to measure those interactions. There was a fascinating meta-study on depression, which revealed in I think a 20-year period, 93% of all of the studies focused on one variable, causal variable for depression at a time, and we're not capturing the interaction. So it has these huge and profound consequences for measurement, and thinking those things through, I think, is really important. Now, I'm not going to pretend to have answers for the very thorny questions you just posed, right? I mean, yeah, we need all hands on deck to try to characterize psychiatric dysfunction, but you know, this is the community that can do that. This is the community who's amazing with measurement and data and defining the dimensionality of a space, right? And so I think we just need more of that quantitative stuff to kind of come in and get interested and compelled, and we need to motivate them. We need to incentivize them as well. Yeah, so thank you for all that.
Dan Handwerker, really great talk. One thing I want to push on that relates to the last question a bit is there's this sort of underlying implication of your talk that if we just understand something a bit better, we're going to come out with this radical new treatment, radical new methods that help people. And that harkens back a lot to the biological mechanisms, and it hasn't changed that much, at least how you presented this. But you know, on the area of treatments, we have treatments that help a lot of people. We don't know the biological mechanisms always, but they help people, and we know they help people. And the trouble is figuring out which treatment is most likely to help someone and getting it to them soon. And with, you know, for this audience, what are the data sets we have that have treatment outcomes are there large data sets? How do we get them? How do we build them so that instead of saying we're going to, you know, classify people into 10 clusters of schizophrenia, we're classifying people into these 5 treatment regimes are the ones that are most likely to affect people in this amorphous, weird category that we call schizophrenia. So how do we get that data set? How do we, you know, communally figure out that's the big data that we need if we really want to help people?
Yeah, thank you so much for that comment. It's a really important one. And for anyone who's interested in following it up, I would also encourage you to read Tom Insel's book, Healing, right? So Tom Insel is the director of the National Institutes of Mental Health for 13 years, and he's famous for saying this quote, I spent 13 years and I funded a lot of $20 billion worth of really cool research, but I didn't move the needle on reducing suicide or hospitalizations. And so Tom really shifted his view and wrote this book on healing about trying to deploy the interventions we already have to people that need them, thinking that that's going to be a faster way to put out the fire. That's how he describes it. And I think that, yeah, that's such an important point. I do hope that it's not an either-or situation. I mean, I know this all comes down to limited resources, but I certainly don't think about this as we're trying to make the decision about, you know, trying to understand the brain or deploying resources better or becoming better social interventions, right? Like, let's rally behind doing both. Let's become partners and do both. Yeah.
And I think it's worth noting, right, that, you know, we talk about, you know, how old is a randomized controlled clinical trial, right? It's actually less than 100 years old. Depending where you look, it's probably about, give or take, about 80 years old. And so for centuries, you could argue for longer than that, you know, we tried things and they worked or they didn't. Now there's a lot of reasons why the RCT is such a, you know, a great development, particularly in clinical medicine. On the other hand, if it gets us too far away from that, you know, looking for the actual, whether it's the mechanism or the reason something works or doesn't. It's a tricky problem to solve. Yeah.
Hi. The first questioner already touched on this a bit, broke the ice for me, so I'm happy about that. But I wanted to put a bit more weight behind it, maybe. But in terms of the predictors of mental health problems, biometric predictors are typically very, very weak, but things like social factors like poverty, early life stress, social support, things like that have very strong effects that you don't really need complex models to see. They kind of stare you in the face. And I think I often see the part of this like multi-level model of genes to neurons to brains is missing like another strata of the social environment and our environment. And I think if we're looking for the fragility of the system, that might be the level where we would get the best bang for buck, maybe. So with the hurricane metaphor, we know that the global climate affects the frequency and the magnitude of hurricanes. So that might be, I don't know, a more beneficial place to look and understand how the brain behaves within this bigger system.
And a great opportunity for me to, as a plug, recommend everyone read a piece that Nicole did that's still, I mean, I'm not supposed to have favorites, but still one of my favorites at the transmitter, which of course I'm the editor of, on weather and the brain, and I just find it fascinating, so please check that out.
Hi. Hi, Alana Jones. Great talk. It actually, one of the reasons why I wanted to come up is to not only express my renewed optimism in the face of the talk, but also to try to think about something that I personally deal with as a scientist, which is thinking about, like, I model things, and a lot of tension that I found in terms of thinking about modeling target systems is that target systems in biology are really complicated. It's the curse of complexity that you mentioned, and a lot of approaches are to assume a simple model, assume a spherical cow. Like, how do you determine first principles first, figure out what's going on, and then maybe add certain things later as you come up with better guesses, better understanding of how the overall system seems to work. But when you consider that we're dealing with more so like dynamical systems which have points of control, I'm curious how we might go about it. In your talk, I saw that you mentioned like starting from something that's more detailed and paring it down. I wanted to plug there's a philosopher who talks about thinking about model systems in abstraction. He's like an ardent opponent of simplicity. His name is Bruce Edmonds, and he talks about this idea of thinking about starting from a descriptive system, an empirically descriptive system, and paring it down to the necessary parts. And I wanted to point this to this idea of thinking about control systems and perhaps connecting, like, to kind of connect two thoughts is thinking about control systems and thinking about them in a system of hierarchies. So the mechanisms by which one system is able to create an emergent central point of equilibrium might be determined by those mechanisms might be other points of equilibrium, and so on and so forth. And I'm curious how, with a shift toward thinking about complex dynamical systems, we might be able to try to connect levels of neuroscience, perhaps, you know, thinking about the MAR levels, like perhaps there's several levels of implementation that have varying, combination of purposes, teleology, computational reasons, normative reasons, goals, the why question, and so on. This was a nice long comment just to kind of state all of that and my optimism in the face of this talk. Yeah.
So thank you so much for that. Yeah, I'll just riff off that a little bit, and I want to not spend too much time because I want to get lots of comments in. But yeah, one avenue along the lines you're talking about that I'm personally very excited about, still I think a little bit speculative, but all of you who deal with recursive neural networks know that when you have neurons like have feed-forward and feedback connections, one of the crucial things to making sure that that neural network doesn't go into a seizure is to balance excitation and inhibition. And I think that that is a wonderful through line there between the question of how does the brain maintain its balance at the molecular level as the brain is continually learning, right? And there's a fascinating homeostatic plasticity and its relationship to criticality, right? That balance that happens in RNNs. I think that's a wonderful, really exciting through line. People are now able to use, criticality-like things to measure things like how sleep restored a brain is in a rat, right? So I think that this is a huge area that it's got a through line that I'm really excited about. Yeah. Thank you.
Thank you.
Hi, my name is Didem, and I am originally an engineer, then turned cognitive neuroscientist, and now back in engineering because of the advent of deep learning. So I had completed a full circle. However, now I am enabled by your talk to talk about the benefits to the society below there. Because over COVID, I had a day job, just a part-time job for recovering myself at a grocery store. And we had training on a terrorist attack, if it ever happens, but we never had training on whether you have a mentally retarded or autistic customer to serve, okay? So I was able to recognize an autistic person from a distance and be able to serve her well. So I want to come to this, I am explaining about this because as scientists or engineers working in the field of this foundational brain research, we never get exposed to the societal interaction, and during our times, we are splitting it up in research and in paper writing, publishing, and all of this has no interaction with the actual society. And expecting to contribute to society without even knowing what it entails is, I think, the big disconnect here. And we need more exposure, and this should be an organic thing, because I had first-hand experience of this during the grocery store job. And I don't know how to fix this, but an eye-awakening thing is that now that in the DSM, autism has a prevalence of 2.3%, and this wasn't like this a couple decades ago. I mean, ADHD is ever increasing, and trying to fix this with more science and more models and more AI, although I'm in that part of the field, now seems wrong to me. I mean, we need scientists to have more exposure to the society and to politics as well. To putting all the onus on science seems now wrong to me. We need to put the onus to the interaction of the scientists with politics. They need to be more proactive, and they need more interaction with the society. This is what I think. Thank you.
A big topic, and one that certainly many people have done a lot of thinking about. You know, it's a complicated one, and I would actually sort of maybe suggest that there's a similar kind of feedback loop that we're looking at. I think that people hear the word politics, and I'm being ridiculously simplistic and reductionist here, but something goes off in their brain that may not be all that positive, and yet that is where a lot of decisions get made. So I think it's fascinating, and conversation is the place to start, no question. Yeah, Andrew.
Awesome talk and discussion. I wanted to bring up this distinction between open loop and closed loop control because it seemed very relevant to many of the things you're talking about, right? So the distinction for those who haven't seen it before is open loop control is where you don't have feedback from the system you're controlling, and closed loop control is where you do, you're measuring something about the system, and you're changing your control based on that, right? And it seems to me that this distinction cuts through some of the therapeutics that we have. Like often psychiatric therapeutics are just drugs that you give to someone sort of open loop. You don't really monitor, at least not very regularly, how they're responding to those drugs. Whereas I think one of the promises of things like cognitive behavioral therapy has always been that they give the patient sort of a kind of closed loop control of the system, like some tools they can use to change when they're in a state that they're not happy about being in, right? And so I wanted to ask if you think, you know, in control theory, we think that for complex dynamical systems, we often need to use closed loop control because we need to monitor the system to know when it's in a state that needs changing and how to change it. And do you think that our sort of medical interventions should also change to incorporate more closed loop control?
Yeah, it's a great question, right? And I imagine it's going to be very case by case, right? Like what kind of complex dynamical system are we dealing with, right? Some just like need a shocker signal, right, to perturb them from one place to the other, and others are going to need, you know, continuous kind of input, and others are going to need reshaping. Yeah, right? So I think there's not going to be a one size fits all, but what's the most promising approach? Certainly, I think in this new era of brain-computer interfaces, both invasive and non-invasive, like the fact that we could even do closed-loop control, it's now possible, like for one of the first times in history. So I imagine that's where some of our big discoveries are gonna be next. Yeah.
Yeah, and I would just want to drop something here because you talked a lot about, not a lot, but you talked about cancer, leukemia as one example. I'm wondering if antibiotic resistance isn't another sort of feedback loop, sort of, it reminds me more of the sort of the rabbit and the lynx, right? Anyway, I'll just leave that, but next question.
Hi, I'm Boaz from the Hebrew University of Jerusalem, and I would like to ask what are your thoughts about the possibility that for certain disorders or current progress state could be less similar to weather and more to astronomy before the heliocentric model, as in we might not need more complex models, but rather rethink our approaches about mental illness and the brain.
Sorry, I missed the part. It's not like the weather, it's like what?
Astronomy before the heliocentric model. Heliocentric model. Astronomy?
Astronomy before the heliocentric model, so, right?
Can you unpack that a little bit more? Like why you might think that?
Yeah, well,
I'm not following.
We're assuming here that our models aren't good enough and we need to be more complex and precise in order to reach this higher level of understanding. But there are certain scientific progresses which don't happen by fine-tuning models, but rather by relooking our assumptions and finding out that maybe a different model from the base.
I see. So you're talking about Copernicus, and you know the sun does not revolve around the Earth revolves around the sun. Yes, no, and I see that. I think that that's one of the characteristics of this shift. I guess I see it as. Sorry, I'm gonna go off here. So I'll rein myself back and just say, I think that thinking up here really helps us understand what metaphor kind of are we applying with. We always start with kind of the type of thing the brain is. And I think the shift from thinking about the brain as a domino chain versus as a dynamical system, I don't think it's just a more complex. I think that that's a Copernican insight in some cases, right? So I think that those shifts are really, really important, and we should think hard about them. I'll stop myself there. Sorry. We can talk offline.
The work that you've done,
Can you hear me?
the work that you've done feels like a gift to early career researchers like myself. I would have loved, when I began graduate school, to have this kind of resource, both in the kind of historical analysis and the scientific analysis, in calling not just for an analysis of what has happened, but strategy for how to move forward. And that, I think, is one of the things that I personally have not seen very often. You saw it in RDoc, you saw it in the 1990s, and exactly what our strategy is today is not something, again, I really would have wished, and I'm grateful that I have it now, and I'm looking forward to reading your book, that I would have had that when I began graduate school. And so I imagine it is a gift to all of us who are beginning graduate school around now. So thank you for that. One of the nice insights, especially that's close to my heart, is in thinking about looking for these control nodes and thinking about the brain as a dynamical system. You open up the opportunity for control mechanisms that are endogenous, using the brain itself to exert control over its own systems. And that is an incredible opportunity that is kind of like, you didn't say it explicitly, but I'm sure it is through and through in your book as well. My question is, what I see very often are really missed opportunities to connect broad, open conversations that we're having within the field around some of these core driving motivational factors. For example, for the past few years, there's been a lot of conversation, a lot of debate around understanding and whether or not neural networks help us understand the brain. And there's a lot of ink and blood and tattered relationships from that debate over the past few years. And I did not hear at any point anyone say in this I could have just been missing something, that understanding should help us control the brain in a way that improves people's health and well-being. If that is our definition of control, which we did not have a definition of control over the past few years, it was just a bunch of aesthetic conversations. If our definition of control was being able to improve people's health and well-being, then what counts as understanding is very different. And so I was curious, my question, what are some of the opportunities that you think we have currently, and the questions that we're currently asking, and the ways that we're currently conceptualizing or understanding the brain, where those of us who care about these issues might want to intervene in the social dynamics in a way that kind of brings people more towards these questions?
So first, thank you. I'm incredibly touched. Thank you for that. And do you have any insight into this one, like the particular issues that there's the natural statement that I think, it's the ancient one. If we want to understand certain types of dysfunctions, it's obvious what functions we should be looking at, right? So if we want to understand depression, maybe we should be studying normal mood fluctuations, right? So that's an obvious coupling. I think for me, one of the biggest insights has been that there was an era in which we kind of needed to oversimplify things to wrap our head around anything, and that it is time now to take on more complexity. To me, if you want to work with dynamical systems, you need a dynamical thing you're trying to explain, right? You can't have a static thing. And so those sorts of things, like how do we get the targets of explanation right? That's the thing this project is going to have the biggest impact on me, is really just changing the targets of explanation and rallying behind things that are more sophisticated than anything I've tried to explain before, to make sure that I don't, because domino chains are so devilishly tempting, and I've spent 30 years with them, right? I'm just gonna get sucked back in if I don't just go after something that's radically different. Yeah.
Thank you.
Thank you.
Thanks. So an earlier question made an important point about precision medicine, and that is that if we want to study individual variation in the brain, it is going to be really important to understand which part of that individual variation is actually relevant to understand the disorder. And one of the major challenges in trying to do that is that there is a very large amount of individual variation in neural measures, also among control participants, among participants who are not diagnosed with any disorder. And so one of the challenges in trying to understand individual variation that's relevant to disorders is that there's going to be a lot of variation that is irrelevant to the disorder, that is mixed with the variation that is specifically related to a disorder. And so there start just now to be some tools to try and separate the variation that is specifically related to a disorder from variation that is in common with the general population. But still, they require very large data sets, and also they typically require categorical diagnosis. So I think one very important direction for the future expansion of this research program is going to be to try to move towards methods that do not require categorical diagnosis and can really use continuous measures. And for that, one key element that seems to be missing is richer and finer-grained measures of behavior and of cognitive abilities. Yeah.
So I want to be mindful of time. This has been a wonderful conversation and will continue to be over not just the next few minutes or the next even couple days as we're all here, but for years and months and years and all of that. So I'm going to ask, there are three people and I want to give everyone a chance, but if we could sort of be rapid fire, if you will, I'm going to start here. Thank you.
Okay. So I'm Usok. I'm working at industry to develop AI-based software that supports diagnosis of Alzheimer's disease and related dementia. So regarding the treatments that you mentioned at the beginning of the talk, I think it is too early to determine whether it is possible to completely treat Alzheimer's or other neurodegenerative disorders, not just slowing the speed of cognitive decline. Because it is because recently developed drugs like lecanemab or donanemab, developed by Eisai and Eli Lilly respectively, are called disease-modifying therapy, but they are just anti-amyloid therapies. So I anticipate the development of drugs that can actually reduce not only amyloid but also phosphorylated tau, because they are mostly more related to the disease. I'm not sure if it is even possible because tau deposition in brain is quite different from amyloid plaques. And also, if you go beyond Alzheimer's, we have to concern several other proteins like TDP-43 for frontotemporal dementia, and alpha-synuclein for dementia with Lewy bodies. So I think we are at very early stage. Probably it will take like decades to see a very great treatment that actually can go disease. Like, so for the research, I think researches are like either too microscopic, like focusing on pathophysiological mechanisms, or too macroscopic, just like scanning fMRI or EEG of subjects. So like, I just want to ask questions about some mesoscopic research, like for instance, studying like circuit level dynamics, which is altered by disease. And I'm not sure if it is feasible, but just like, yeah.
Thank you.
Thank you.
Take very quickly a couple more. Yeah.
Of course. Science is sort of, in some ways, it's sort of a myopic process. And I'll also emphasize that as a graduate student, I will repeat the comment of the last commenter that this type of high-level discussion is a gift. Science, in some ways, is kind of myopic in that we take this dynamic programming approach where we have problems that are split up into smaller and smaller problems. Individual scientists design experiments where your goal is to create extremely tight feedback loops so that you can study just one aspect of some complex dynamical system. And so history, like the history of science is kind of the history of either overly simplistic models or, you know, another commenter mentioned the Copernican paradigm shift. How do we keep the grand plan in mind when we're teaching science? And we're trying to teach people about creating these really tight feedback loops so that you can observe causality, but also keep in mind that systems are very, very complicated, and that what we learn from tight feedback loops isn't necessarily completely generalizable.
Question. I'd love to organize, hash it out. I think that's a great conversation.
I'll be brief since I know we're short on time. So my name is David Tovar. I also did medical school and went full into research after medical school. And one of the biggest things that I see is just data in terms of the documentation that you get from physicians, you know, even a given patient trying to see notes from an outside hospital is impossible. And so one of the things that I see as the biggest issue in terms of applying all of these great models that we have, and also a great opportunity, is being able to collect the data, make documentation easy for physicians. We have a host of physiological data that we collect that goes nowhere. If you start correlating that with EEG, fMRI, and all the things that we collect, you start making real progress. And so that's my primary comment. Thank you.
Thank you. Well, thank you all. Thanks for those fantastic comments and questions. I just also want to recommend, I don't know if we can quite pre-order it, but soon enough, do look for, I'm sure Nicole will post all kinds of news about her book, which I'm really looking forward to. In the meantime, you can follow Nicole's work, obviously, in many ways, including at The Transmitter, where she contributes regularly as a contributing editor, which we're so thrilled about. So let's just all thank Nicole for a fascinating discussion and leading the moderation. Thank you.
As well as Ivan. Thank you all for coming.